Hi everybody. Today we are, or at least in this video, we are going to talk more about linkage, and we are going to start with an organism with this genotype. However, the genes are linked and the alleles are arranged on the chromosomes as indicated here. So maybe you remember from the last video that this notation is useful for indicating the arrangement of alleles on chromosomes. So if these are two chromosomes, homologous chromosomes, in the individual with this genotype here, this notation here is saying the big A and big B allele are on one chromosome, and the little a and the little b are on the other chromosome. Now, just like we have seen before with an individual with this genotype, we can get 4 genotypically distinct gametes produced by this individual. And again, it doesn't matter if it's mouse or flies or frogs or humans. We can get 4 types of gametes here, or genetically distinct gametes. And the 4th one is right there. So we have big A, big B, little a, little b, big A, little b, little a, big B. Now these right here are the we're going to call them the non-recombinant gametes. And you'll see if you're looking at the notes, sometimes I call these non-crossover gametes. So we can kind of use these words interchangeably. So for now, I'll call them non-recombinant gametes. And these right here, we're going to call these recombinant gametes. Now, a lot of students have a common question, and that is, how do you know which ones are the non-recombinant gametes and which are the recombinant? I didn't just pick these at random. So there's a method that you can use. So take a look at the way the alleles are arranged in the parent up here, or in the individual. So we have big A and big B on one chromosome, little a and little b on the other chromosome. So this gamete here is inheriting this chromosome here with a big A and a big B allele. So the chromosome is, exactly as it is found in the parent. There's been no changing. There's been no recombining of alleles. So the big A and big B are still next to each other on the chromosome. So, and same thing for the little a and the little b. So little a is here with little b in this gamete with one of the homologous chromosomes. And you can see in the parent, little a is with little b. So there's been no changing, no recombining of alleles. So these are the non-recombinant gametes. However, down here, so what happened down here? Essentially, what we're saying is now we have big B with little a here. So this gamete has this chromosome where the alleles have been changed. So this is a recombinant chromosome, and thus this is a recombinant gamete. So little a is with big B. That combination is not present in the parent. So this must be a recombinant gamete. Same thing for this one. Big A is with little b. So I can diagram what that would look like after the recombination, which we'll talk about later. So this is the recombinant gametes because big A is with little b. This arrangement does not exist in this parent here, normally in the diploid cell. So only during meiosis and formation of the gametes do we get the big A with a little b. And we don't get that all the time. So as we're going to see, most of the gametes that are produced by this individual right here will be non-recombinant gametes. a certain fraction of the time, we'll get recombinant gametes produced. So our first question is, what is the ratio? What is the of all the gametes that this individual will produce in its lifetime? You know, what is the ratio of non-recombinant gametes to recombinant gametes? Or if you were to take 1,000 gametes produced by this individual, how many would be non-recombinant and how many would be recombinant? So we're going to see how to calculate those numbers and learn a little bit about linked genes and what we're going to call a genetic distance between the genes. So our question here, we want to know in this individual, what is the ratio of non-recombinant gametes to recombinant gametes? And how are we going to figure that out? Well, we're going to do a genetic cross because this is a genetics course, sorry. So we are going to take the parent with this genotype, and we're going to diagram the gametes down here. And remember, our question is how many of each of these gametes is likely to be formed in this individual. So in order to do that, well, we're going to need to actually count this somehow. We can't count this directly. What we need to do is we need to infer the number of gametes produced by this individual by doing a genetic cross and looking at the phenotypes of the offspring. So we are going, in order to figure that out, we are going to need to mate it with another parent, parent number 2 here. And we are going to have to be careful with the genotype we select for parent number 2. So in this course, it is almost always going to be the homozygous recessive genotype. I think we will look with problem 5.18, a case where that is not the case. But usually you will be safe if you choose homozygous recessive for both genes for the mating partner when you're trying to figure out the different types or numbers of different types of gametes in parent 1. So I'm going to put AB there and AB here. And so what you want to do is we need to choose a genotype that allows us to connect each parent 1 gamete genotype, so each of these, with a unique offspring phenotype. So choose genotype for parent 2 that allows us to connect parent 1 gamete genotype with a unique offspring phenotype. Okay, so, and I already told you that this one right here, homozygous recessive for both gene A and gene B, is going to allow us to do that. So let me show you why that is the case. So this is the genotype of all the gametes produced by this individual right here. All of them have to have a little a allele and little b allele. So for the offspring, we are going to have 4 possibilities or 4 ways to produce an offspring. So this gamete can be combined with this one, this can be combined with this with this, or this with this. So 4 different types. So I'm just going to list these guys right over here. So these are the genotypes. And I put little numbers here. So these are the genotypes of the alleles from parent number 1. And then I can just put the alleles from parent number 2 underneath. Okay, and so we're showing the arrangement. So it inherits so each of these offspring inherits one chromosome from parent number 1 and one chromosome from parent number 2. The chromosome from parent number 2 is always the same, little a and little b allele. The chromosome from parent number 1 changes in each case. So these are the genotypes. What about the phenotypes? Let me put genotype right here. Genotype. Now phenotype, I'm going to list that over here. So each of these has a phenotype, and I didn't define traits for each of these, like plant height or eye color in Drosophila or fur color in guinea pigs. We're just referring to generic genes and generic traits, and the alleles are completely dominant or completely recessive here. So with respect to phenotype, we can say this individual right here has the big A and big B phenotype. This one here has the little a and little b phenotype. These are recessive traits, and they have 2 little a alleles, 2 little b alleles, so little a, little b. This one here, big A, little b. This one here, little a and big B. Okay, so this is a good time to introduce 2 terms. These right here, these phenotypes, we can call these parental types. So these offspring have parental type phenotypes. What do I mean by that? So take a look at parent 1. Parent 1 has the big A, big B phenotype. Parent 2 has the little a, little b phenotype. So these offspring right here have phenotypes that match one or the other parent. So these down here are recombinant types. Neither phenotype matches the parents exactly, right? So this one here has the big A phenotype, little b, and this one here has little a and big B. So these are recombinant types, which makes sense. As you will see, these are the phenotypes that were produced as a result of recombination in parent 1. They came from the recombinant gametes. So let's just make sure, as we said before, we need to choose a parent 2 genotype that allows us to connect each P1 gamete genotype, these guys right here, connect each of these with a unique offspring phenotype. So essentially, we need to be able to look at the offspring phenotype and know what the genotype of the gamete from parent 1 is or was that led to this. So by looking at this, we should be able to infer the gamete genotype from parent number 1 that led to this offspring. So this is little a and big B, and we can see, here it is. Big A and big B. We can see, yep, okay, this was the chromosome that came from parent 1, big A, big B. This one right here, little a, little b. Here it is right here, little a, little b. Big A, little b, big A, little b, little a, big B, little a, big B. Okay, so we're perfect. So everything is working out. And now if we have numbers for this, if we do this cross and we get the offspring and we count up the number of offspring with each of these phenotypes, we know the number of each of these gamete types here that were produced by parent number 1. So let me neaten this up for you here. Now let's say let me address one thing actually. What I want to do first is, before I give you numbers for these guys, I want to address where these recombinant types are coming from, okay? And when they're not recombinant types, where do these come from? And we've seen this before in the first 2 lectures of the course. We talked about meiosis and tetrad formation and chiasma, but now we're going to see how the chiasma actually leads to genetic variation in offspring. So we're going to have two types of tetrads here. So one tetrad is going to be let's call this a non-crossover tetrad. And this tetrad over here is going to be a crossover tetrad. And we're going to diagram the example in the heterozygous individual, heterozygous for two genes. And when we talk about non-crossover and crossover, we are referring, we're talking about these with respect to these two genes right here. So in one tetrad, we have big A and big B on the sister chromatids, and that's just one of the chromosomes of the tetrad. And I'm going to draw the chiasma way down here, or crossover. So chiasma is what they called them when they were first studying meiosis, and they saw these points where the tetrads, the sister chromatids were overlapping. We called this a chiasma. We now know it's a crossing over point. It's a point where there's actually a physical exchange of the DNA in this chromatid over here with this chromatid over there. Or vice versa, and vice versa. So, okay, so in this tetrad with no crossover between A and B, all of the gametes are going to have big A, big B, or little a, little b genotypes. So they are all non-recombinant gametes. Okay, so how did I get this AB? Well, I like to just start at the top, see A, B, follow it down, and then there we go. For this one, A, B, follow it down, okay, A, B. And then with this point right here, I just turn back this way. So it doesn't really matter how you trace the genotypes with these sort of diagrams or trace the chromatids, as long as when you look down here, you know, you get, okay, all non-recombinant gametes, okay? But over here, now let's look at the crossover condition where we see a crossover between A and B. So big A, big B, big A, big B. Now I'm going to put the chiasma right here between A and B. Now we have the little a over there and the little b over there. And here's the tetrad, the two chromosomes here. Remember, these are the sister chromatids right here. So two pairs of sister chromatids. Now, okay, we see the crossover here. So after this goes through meiosis, the gametes are formed. We are going to have a big A and a big B here. I'm going to follow it down like this. And see this A, this little a, I'm going to follow that this way. When I get to this point, I'm going to go this way, and we got a big B. Now over here, big A, I'm going to hit the crossover point and come back towards this chromosome. Big A, little b. And this one, A, B. Okay, so we have, in this condition here, we have the recombinant gametes formed, where there's been an exchange of alleles between the chromosomes. Now here we have a non-recombinant, and here we have a non-recombinant. So even, this will come back later, and maybe I think the next video, even when there's a crossover in the tetrad between A and B, half of the gametes that are produced are non-recombinant gametes. So, okay, so this is where the recombinant gametes are coming from. There's a crossover, and, you know, this doesn't happen all the time, and the frequency with which it happens depends on how closely related, how closely the gene A and gene B are to one another. The further apart they are, the more likely you are to see a chiasma form between the two genes and to get recombination between those two genes. Now, let's go back to our example where we were able to count, or we were able to do a cross and see a bunch of offspring phenotypes, and we were able to correlate those with, this was parent 1, with gamete genotypes. So let's say 498 of the offspring had the big A, big B phenotype. Well, that means they were produced from 498 of these big A, big B gametes here. Same thing for these. Let's say there were 497 offspring with little a, little b. That means they resulted from, 497 gametes that were little a, little b. And for here, let's say we had 2 and 3. We already determined that these are the non-recombinants. These gametes are the non-recombinant gametes, and these gametes are the recombinant gametes. Now, we can use these numbers to make a genetic map of the chromosome, at least with respect to genes A and gene B. So I'm going to put gene A right here and gene B right here. And what we need to do is determine what percent of all the gametes made by this individual were recombinant gametes. So if you add these up, they should add to 1,000. And 5 out of the 1,000 were recombinant gametes. So 5 divided by 1,000 is 0.005. So we multiply that by 100 to get the percent, and that gives us 0.5%. Okay, so what we're going to say now for this little genetic map, we're going to say these are 0.5 centimorgans apart. And this is a genetic distance. And what is a centimorgan? So a centimorgan, essentially, 1 centimorgan equals 1% recombination, or 1% recombinant gametes. So that's how you determine genetic distance between two genes. You do a cross, you determine the number of gametes produced that are a result of recombination. You determine, said another way, you determine the percentage of recombinant gametes that were formed. And well, that's the genetic distance between gene A and gene B. So genetic distance is or equal to percent recombination. That is different from physical distance, which is the number of base pairs between 2 points on a chromosome. So the physical distance would be the number of base pairs between gene A and gene B. The genetic distance is the percent recombination between gene A and gene B. Now, why do we have two different measures? Well, for one reason, genetic distance was, this calculation came about before we even knew the structure of DNA, before we even knew what, a base pair was. So genetic distance sort of comes way before, maybe 30 years or something before physical distance measures became possible. And today we care about genetic distance because there should be, overall, there's usually a correlation between genetic distance and physical distance, where the longer the genetic distance, the longer the physical distance. Sometimes that's not the case, and there are a lot of scientists that spend a lot of time trying to figure out what is it for those rare cases where there's no correlation between the two. What is it that causes, say, genetic distance to be really high if physical distance is low? For the most part, they are correlated, though. Genetic distance correlates with physical distance. If this is larger, then this will be larger. So for this part of the course, we're going to be mostly concerned with genetic distance. And when we move into molecular genetics, we will move towards physical distance. Okay, so far in our example, we learned how to determine the distance between two genes by performing a genetic cross. Now, there's some other things we can do